Hi, Year 12s, welcome back. So this video is going to be on DNA amplification, PCR, but it's also going to mention some enzyme stuff at the start. So we're going to go straight into it. Okay. The enzyme things is mentioning competitive and non-competitive inhibitors. All right. So just sort of finishing off the enzyme stuff that we were looking at. So enzymes can be hindered by molecules known as competitive inhibitors. All right. And they impede enzymes by blocking their active site. So that's your competitive inhibitor here, this orange molecule. All right, that comes in and that will sit right there in the active site. All right, it will sit there and it'll block these two substrates from coming in, so they can no longer do their job. Okay. And therefore it's competitive because it gets to the site first and it stops them from entering the active site. Okay. So no products are created. Second type of inhibitor is a non-competitive inhibitor. It doesn't compete for the active site. It goes the backdoor way. It finds this allosteric site here. Okay. In this allosteric site, it will then slot in, as you can see here, and that will deform or change the protein. Okay. So it'll change it into a different shape. It'll change that active site. Okay. And therefore, the two substrates can't come in and get turned into products. So no catalyzation, no enzyme doing its job. Okay, we also have cofactors and coenzymes, which also exist. So some enzymes require assistance from a cofactor to catalyze their reactions. Cofactor is a molecule that assists in enzyme functioning. All right, coenzymes are a subset of cofactors. Subset. So coenzymes are made up of cofactors, but they're organic, non-protein molecules, and they're used within the reaction. So they're used up, okay? Unlike the actual enzyme, which can repeat the process, okay? Depending on whether it's catabolic or anabolic, it can repeat the process. Alrighty, so we'll move on to PCR. So what exactly is polymerase chain reaction? It's a DNA amplification technique. Key word here, amplification. You have a little bit of DNA, you do something, eventually you get a lot more DNA. Okay? And we have a hell of a lot over here. That's all it is. Alrighty. So what is it used for? It's used for paternity testing, so finding out the father of a child. It's used for specifically for forensic testing, samples of bodily fluids and different things. All right, so it could be fluid, it could be hair, it could be anything with DNA in it, skin, whatever it is, blood. All right, as long as it has DNA in it, we can use PCR and then try and find the person, all right, or match it to a suspect's DNA. It can also be used for analyzing gene fragments for genetic diseases. All right, that'll come back to us in Unit 4. So that's all the things we need more DNA for. All right, so we're going to start off by talking about primers. These are made in this here machine, okay, and that's the DNA synthesizer, and it's really a printer for DNA, okay, and this will come back. So when we look at PCR, this will come back because we need primers, and this is where the primers are made. Okay, there's a few bottles here that contain a bunch of different reagents and things that allow the primer to be created. All right, so PCR, it occurs on small lengths of DNA usually, so 100 to 10,000 base pairs long. It's used to replicate the specific sequence repeatedly. All right, so there's three main steps to this process. There's denaturing, which we've heard of before with enzymes, obviously. But in this case, it's a nucleic acid, it's not a protein. So the denaturation process doesn't entirely destroy it. And in this case, it's not irreversible, it's actually reversible because it's a nucleic acid. All right, we have annealing, which just means something sticking. And then elongation, also known as extension, where the polymerase molecule will run down the strand. And it's a specific polymerase that we're going to talk about in a sec. Alrighty, it's the specific polymerase is this Taq polymerase. Alright, thermus aquatis polymerase. So T.AQ, I guess you would say. Alright, and that comes from a particular bacterium, which is a thermophile. Okay, so it's an extremophile, so it can live in extreme places, and in this case, it's for temperature, so thermophile. All right, so we're going to talk about thermal cyclers. This is another piece of laboratory equipment, and essentially that is like an oven and a freezer put together that works really fast on DNA. Okay, so it changes the temperature. So pretty much they add a few different things in, and they make a big soup to do this PCR process. All right, what are the things that go in? Firstly, a buffer to keep the pH. All right, if you're making a bunch of nucleic acids, obviously that's going to get acidic, so you need the buffer to stop that. Keep it a bit more regular. Mixed with obviously a few other things. So we have the DNA, or the target DNA gene that we're looking at, the primers which have been made in that other machine that we looked at. Obviously the Taq polymerase, Taq polymerase protein that needs to run down the strand, magnesium chloride, and of course DNA nucleotides to create the DNA. The second part of this, and probably the most important, is these temperatures here of which each step occurs. So step 1, if you remember, was of course denaturation. All right, so the DNA is denatured or melts because of the heat, and it breaks the H bonds between the complementary nucleotides. All right, and that occurs at around 90 to 95 degrees Celsius. All right, so that's step 1. Step 2, obviously at 55 degrees here. Okay, so a lot cooler. That's the primers annealing to the DNA. So 55 to 60 or even 50, that's where the sticking of the primers to the DNA is. And the primer is where the Taq polymerase will bind. Okay, it's similar to when we looked at the trip operon, and we looked at RNA polymerase binding to the promoter region. Okay, and then lastly, number 3, and the most important step, and you do need to know this specific number, so 72 degrees Celsius, that is where our boy Taq polymerase can do its work. Right, it extends the primers because 72 is its optimal temperature, and then from there you get your new DNA strands. All right, again, PCR requires DNA sample, Taq polymerase nucleotide base. So this allows Taq polymerase to essentially attach and begin extending the DNA strand with of course, the primers and the nucleotides. And yeah, that's pretty much PCR for you. Okay, so that's enzymes and PCR. Like and subscribe. See you in the next video. Strand.